Hello everyone, it's Mr. Wolf here, and in this video I'm going to be going over the formative specifically for experiment A. All right. Do germinating versus non-germinating seeds do aerobic cellular respiration at different rates? All right, so the independent variable here is going to be the type of pea. This is what we can manipulate. So specifically, we're either going to use germinating peas or non-germinating peas. All right. And we're trying to see the effect on aerobic cellular respiration. So I could either measure the reactants, or I can measure the products. And luckily we have the CO2 sensor and the O2 sensor. So my dependent variable is either going to be the amount of oxygen or CO2. So specifically, how am I measuring that? With the oxygen or CO2 sensors. All right, and it's important to also include units as well. So for oxygen, it's going to be the percentage of oxygen, and CO2, it's going to be in parts per million. So with that in mind, let's actually look at the experimental setup that I drew here for that. So again, independent variable, germinating peas versus non-germinating peas. All right. I'm using the oxygen or CO2 sensor, and then I'm also including other constants and parameters. So I said in this experiment I'm going to test 15 peas each. You might have had a different amount. I said I'm going to use the same temperature, same type of flask, and then the same time I'm going to record for 3 minutes. That's my parameter. Okay? All right, so let's look at, by the way, as you guys are watching this video, make sure that you are in a different color pen, kind of giving yourself feedback, whether it's kudos or whether it's some fixes. All right, so in this case, did you include a control group in the diagram above? It could have been yes or no, right? As long as you defend your reasoning here. So if you circled yes, you may have said that you know what the normal condition is dry peas. That's our control group, all right? And I'm fine with that. Right? I'm guessing maybe a lot of you said that no, there's really not a control group because there's no like normal condition or no group that doesn't receive the treatment. However, we could still compare germinating to non-germinating peas, so we're good, right? Because the whole purpose of a control group is to compare one group to another to see if there's a difference. All right. So constants in this experiment. You may have hit some different ones, but I said a few. Same amount of peas, the same temperature, same type of beaker, right? I had drawn a lot of that right here in my drawing, right? So make sure you're specific when you're drawing your setup here. I also said the same same span of time of data collected for, so like 3 minutes, for instance, and then tested at or around the same time. All right, some additional parameters. So these a lot of times students will miss. So what I'm looking for parameters is give me some specifics, right? How are you specifically going to run your experiment? And you guys are going to run this on Friday. So in this experiment, I'm going to use 15 Ps, is what I said. I'm going to run 3 trials. You could have said 4, 5 trials. I'm going to collect data for 3 minutes, right? Again, you may have had different specific parameters, but as long as you have specific parameters, you can't just be vague about it. All right, so let's look at our data table now. All right, so a few things. I'm actually going to cover this up on purpose, all right, because a lot of the times students will take this title, like the effect of germination on the rate of cellular respiration, and they'll put it right here where my pen is. All right. So overall, this is a pretty good table, right? Here's my independent variable in a column, and then I specifically say whether it's going to be my non-germinating or germinating peas. I have multiple trials. I could take an average, which is great. But let's say that I put a number in here for, what does that for mean? For what? Right, so that's why it's important. You have to include how you're measuring, right, whatever you're measuring. So that's your dependent variable. So you could have said the amount of CO2 in parts per million, and you make sure that you include your units, or you could have said oxygen in percentage and make sure to include your units as well. All right, so this has been a pattern that I've seen of things missing from a data table, and you have to have this to have a sufficient data table. All right. So then let's look at the graphs. The graphs, you'll see 2 right here. You could have actually had multiple graphs for this. All right. So let's just go to the reasoning right here. All right. You could have had a bar or line graph depending on what you decided to make your x-axis. Just remember, we always use a bar graph if the x-axis variable, usually our independent variable, is going to be in categories, right? Germinating peas versus non-germinating peas. All right. But we use a line graph if our x-axis variable is continuous. So for instance, like time. Right? When we actually collect data, we're going to use a graph like this. All right? This was the one, if you remember in class, when we tested out the germinating peas and we saw oxygen going down, and we kind of had a graph like this. All right? And then what we could do is we could differentiate between germinating and non-germinating peas by making a line graph and having two different colored lines. Right. On the y-axis, regardless of what graph we make, that's always what we're measuring. So this is our dependent variable, and in this case it could have been the amount of CO2 in parts per million, make sure to include your units, or the amount of oxygen and a percentage. Again, make sure to include your units. All right. Even though we're going to use this type of graph, my guess is most of you made this one, and that's fine. That's fine for this formative. Okay. So again, dependent variable, the amount of CO2 in parts per million, amount of oxygen in percentage, and then looking at germinating peas versus non-germinating peas. All right. Now, one thing, if you look at my key, is I actually missed something. I missed the title right here. So something along the lines of, for the bar graph, similar to what my title is up here, the effect of germination on the rate of cellular respiration. And I could have also had a similar title right here as well. And I could maybe have added on like the effect over time, right, since I'm doing a line graph with time right here. All right. So if you have any questions after looking at this key, what I'd like you to do is on like a piece of paper, hopefully a note card that you could get, write down the question you have, write down your name, turn it into the sub, and I will make sure to look at it and get back to you personally tomorrow. Thanks for watching.